Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the topic is related to immunology, and the title of my today's lecture is Phagocytosis. And the main concern is that how microbes are recognized by phagocytosis. This lecture consists of two parts. In the first part, in this part, I will discuss that how the phagocytosis process begins and how the microorganisms interact with the immune cells. So after that, when it is interacted and there is a positive signal, and after that, these microorganisms are engulfed by the immune cells. And the next stage is that it is then digested, that is known as the intracellular digestion. And after that, these microorganisms are, when they are killed, so they are exocytosed, means they are released by their immune cells. So in these video lectures, it consists of two parts. In the first part, I will discuss that what are the different receptors that are present on the immune cells and how they interact with the microorganisms. Okay, and in the second part, I will discuss the intracellular digestion, when the microorganisms are engulfed by the immune cells, and after that, when they are destroyed, so how they are released into the environment. Okay, so watch both the videos so that you are much clear about the phagocytosis process. Okay, so let's start with this video. First of all, keep this thing in mind that we are having three types of cells that are known as phagocyte. Okay, that are known as phagocytes. Okay, the first one is the macrophage. Macrophage is a phagocyte, and it is largely used in phagocytosis process. So macrophages engulf the microorganism and destroy it, and then release it into the environment or present it or itself. And the second one is the B cells. Okay, the first one is the macrophages. They are also known as phagocytes, and these are also known as macrophagocytes because these are large in size. And the second one is the dendritic cells. So dendritic cells are also phagocytes, and these are also involved in high amount of phagocytotic process. Okay, the third one is the B cells. B cells are also they are also performing phagocytotic process. Okay, so these three cells are mainly involved in phagocytosis. The fourth cell I have not included here, but the fourth cell is the neutrophils. Okay, the first one is the macrophage, the second one is the dendritic cell, the third one is the B cells, and the fourth one is the neutrophils. Okay, neutrophils are also known as microphagocytes. Okay, these are also known as microphagocytes. Okay, so keep this thing in mind. If someone asks you that which cells are used in phagocytotic process, so your answer will be that macrophages are also included, dendritic cells, B cells, and neutrophils are the cells that are involved in phagocytotic process. Okay, so this was a simple information. So first of all, keep this information in your mind. Okay, I will take macrophage as a model cell. Okay, although all the receptors that are present on the macrophage will also be present on the dendritic cell, on the neutrophils, and the B cell. So I'm taking macrophage as a standard. Okay. Okay, if we proceed further, so this is an overview of phagocytosis. I'm not discussing it here. I will discuss each and every in detail. So first of all, this is this is a phagocyte. This is, for example, this is a macrophage, and these are the bacteria. Okay, so these are engulfed. First of all, these bacteria interact with these immune cells, these macrophages. After that, when there is a positive signal, so they are engulfed, and in this process, in this C stage and D stage, this stage is known as the intracellular digestion. So these microorganisms are digested, and after that, when they are killed, so they are released to the environment. Okay, so in this video lecture, I will show that how these bacteria interact with these immune cells, and in the second video, I will discuss that how this intracellular digestion is processed. Okay, and at the last, this E is the exocytosis. So how different immune cells release the microorganism to the environment, the debris of microorganism to the environment. Okay, so this is a simple overview. Each and every step I will discuss in detail. Okay, now come to the receptors. Okay, that how microorganism interact with its this immune cells. Okay, for example, this is a macrophage. Okay, as I have told you that I will take macrophage as a standard immune cell. Okay, so the first one, if I start from here, so the first receptor is known as the RGD receptor. RGD means R stand for arginine, G stand for glycine, and D stand for aspartate. Okay, these are the amino acids. So if I want to tell you, so microorganisms, if we look to the structure of, for example, this is a microorganism, this green. Okay, if I write M. Okay, so this is a microorganism, and as we know that it consists of a cell membrane, a cell wall, and there are different proteins present in the cell membrane and in the cell wall. Okay, so we all know that proteins are composed of amino acids. Okay, a variety of amino acid. So this receptor, what does it do? It detects the arginine, glycine, and aspartate amino acid of the protein present in the present on the surface of the oh, sorry. Okay. Sorry for the interruption. So these these receptors, these yellow color is the receptor, and this is the receptor that detect the arginine, glycine, and the aspartate amino acid of the proteins present on the surface of the microorganism. So when these amino acids are present in the protein portion, so what will it do? It will interact with this receptor, and after that there will be a positive signal. Okay, there will be a positive signal, and these microorganisms can be engulfed by this macrophage. So these are known as the RGD receptor. Again, I tell you, R stand for arginine, G stand for glycine, and D stand for aspartate. Okay, these are amino acids present in the protein. The second receptor is that the PAMP, and this is known as this is an abbreviation. PAMP stand for pathogen associated molecular patterns. Okay, and so these and the receptor present over the immune cells is known as the PRR. PRR means pathogen recognition receptor. Okay, pathogen recognition receptor. So as I have one thing I want to tell you guys, that there are certain for example, there are certain molecular patterns that are present on that are specific to each and every microorganism. For example, here is a bacteria. It consists of a flagella. So flagella is a unique structure of a microorganism of a bacteria. So these can be known as the pathogen associated molecular patterns. Okay, flagella consists of a specific molecular pattern. So these patterns are detected by the pathogen recognition receptors, and after that when it gets a positive signal, so these are engulfed. There are other such type of pathogen associated molecular patterns, just like pili of the bacteria, the capsule, the molecular capsule, the capsule, the slime layer. Okay, these all are pathogen associated molecular patterns. The lipopolysaccharides, the lipotecoic acid, the teichoic acid, these all are specific to each and every microorganism. So these special patterns are detected by the pathogen recognition receptor present over the immune cells, the macrophages, and when they are detected, so there is a positive signal, and after that these are recognized and these are engulfed by the immune cells. Okay, so this is the second one. The third one is the flagellal lectin. Okay, this is also a type of receptor. This is as we know that there is a lectin is a special type of protein, and it is mostly present in the flagella of the microorganism. Okay, so this lectin is detected by a receptor that is basically a carbohydrate receptor. Okay, So these carbohydrate receptors can only detect the lectin of the microorganism that is present in the flagella. So it is detected, and when there is lectin, so there is a positive signal, and the microorganism is engulfed by the macrophages. Okay. The last one, the fourth one is the hydrophobic interaction. Okay. Hydrophobic interaction, if microorganism interact with the immune cell through hydrophobic interaction, so it can be engulfed by the immune cell. What is hydrophobic interaction? The interaction between the fat molecules and the water molecules. Okay. So as we know that most of the bacteria consist of slime layers and capsules, and there is a water content in that slime layers. Okay. So the watery part of the slime layer interact with the phospholipids of the immune cells. Okay. So when it is enter, there is a slight interaction in between them, and after that, when there is a positive signal, and these microorganisms are engulfed by the immune cell. Again, I repeat that what is hydrophobic interaction? The interaction between the fat molecules and the water molecules. Okay. So the water molecule should be either from the immune cells or from the microorganism. If we consider the slime layer, so slime layer consists of high water content, so it interacts with the watery part, or sorry, it interacts with the phospholipids of the microorganism of the immune cells, and after that there is a specific interaction, and these microorganisms are engulfed by the immune cells. Okay. So these are the four type of receptors, and these are the four type of interactions. Okay. By which the immune cells can get their microorganisms and engulf them and digest it and kill them. Okay. The fifth, there is also a fifth type of receptor, and it is very much important. That receptor is very much important. Hope you have understand these four receptor. Now this is the fifth receptor, and it is very much important for each and every immune cells. Okay. And these are known as the toll-like receptors. Okay. Oh, I think you have known about this. You have heard this word, toll-like receptor, TLR. Okay. So on macrophages, on dendritic cells, on B cells, on neutrophils, there are toll-like receptors and a variety of toll-like receptors. If you look here to the diagram, so there is TLR1, TLR3, 7, 8, 10, 9, 5, 4. There are about 20 to 40 different toll-like receptors. Okay. I am confusing this. I will verify it, and then I will add in the captions that how much toll-like receptors are there. But I think there are 20 toll-like receptor, different type of toll-like receptors. Okay. So what does they do? These each toll-like receptor is specific for a specific pattern. Okay. If you look here to the toll-like receptor, this TLR4. Okay. If you look here to the TLR2. Okay. Toll-like receptor 2. So it can bind with the bacterial lipoprotein. Okay. It is performing a receptor for the bacterial lipoprotein. So these, I mean, it is interacted with the bacterial lipoprotein. So after that, these bacteria can be engulfed. If you look here to the TLR6. Okay. So TLR6 interact with the lipopolysaccharide of the bacteria, and after that, when it is when there is an interaction, so there is a positive signal, and this bacteria is engulfed. If we look here to the different other receptors, so if there is if we look here to the TLR9. Okay. So TLR9 detect the bacterial DNA, and it detect the cytosine phosphate guanine. Okay. It detect the bases that is specific with cytosine and guanine. Okay. And after that, when it is detected, so this TLR then give a positive signal. Okay. It give a positive signal to the inner cell, and after that, these are engulfed. Okay. If we look here to the other type of receptor, there is also, so TLR is the fifth type of receptors. This is a fifth type of receptor. And there is another type of receptor that is known as the look here, if we look here to the TREM1. This is the sixth receptor. Okay, so TREM1 is also specific for the lipopolysaccharide. TLR4 is also specific for lipopolysaccharide. Sorry, TLR6, but the most specific is the TREM1. It is also specific for lipopolysaccharide. The seventh receptor is the mannose receptors. Okay, we all know that in fungi there are manoproteins in its cell wall. Okay, so these mannose receptors detect the mannans of the fungi. Okay, because there are manoproteins on the surface of the fungi cell wall, so these are detected by the mannose receptor. So these are the seven. This is the seventh receptor of the that is present on the immune cells, the macrophages and the dendritic and the B cells and the neutrophils. Okay, so at the last, when a summary, if I want to tell you, so what does it do when these all give a positive signal? So it can also engulf the bacteria, and plus there are special type of transcription factors. Okay, there are special type of transcription factors. What does they do when these immune cells are activated? So signals are produced, and these signals activate the transcription, different type of transcription factors. Okay, and these transcription factors then induces proteins and transcription of the DNA, and different proteins are produced in the form of cytokines. Okay, in the form of cytokines, and these cytokines are then released by the immune cells, the macrophages, and they interact with the adaptive immunity. So what does again, if I want to tell you, so when these interaction is successfully conducted, so there is a positive signals and different transcription factors are activated. And transcription factor, what does they do? They are, each transcription factor is specific for a specific transcription, for a specific transcription of a protein. Okay, so what does they do? They induce the transcription process, and different genes are transcribed and different proteins are produced. And if I want to name a specific protein, so cytokines. So what does they do? The cytokines are specific proteins that are involved in signaling. So these signals are then passed to the adaptive immunity cells and different other cells to tell that there is a certain microorganism and should get alerted. Okay, so this was all the seven receptors that are present on each and every immune cells of the macrophage, dendritic cell, B cells, and the neutrophil. Okay, and this is a summary in this table. This is a summary of these receptors that I have discussed: the lectin carbohydrate receptor, the protein receptor, hydrophobic interaction, and the signaling. And this is an informative slide, so you can just compare it with the TLR1 and TLR2 detect the bacterial lipopeptides, and different other TLRs detect these certain type of proteins. Okay, so this was all about that how microorganisms are recognized by phagocytes. Okay, in the next video I will discuss that. Now after that, when these are recognized, so these are then engulfed. Okay, so in the next video I will discuss that how these are then destroyed by intracellular digestion. Okay, what happens inside the macrophages to the microorganism, and after that, when they are destroyed by different chemical reaction, I will also show you the different chemical reaction by which the microorganisms are killed. Okay, the mechanism. So when they are killed, so then how they are presented, and how are they released to the environment. And this is a topic for another video. I hope you have understand this video. If you have any question in your mind, so you can ask me in the comment box. I will answer you there. Okay, and if you have understand this video, so like this video, share it with your friends, and subscribe to my channel for more interesting videos related to science. Thank you very much. God bless you.